So last time we discussed the earliest examples of small-scale systems biology. One was a toggle switch, and the other was refrigerator, published in the same issue of a journal 13 years ago. And then I moved actually a bit closer to the present time to discuss a single-cell measurement of the gene regulation function done by Elovich. And there, the function that they use to fit the data, I remind you that gene regulation function is basically what is the production rate of the protein gene product of your interest as a function of repressor concentration in the cell. Okay? And that function that they measured in the cell could be fit with the form of equation that looks like this. Basically, that is the concentration of the repressor that you are dealing with, and there's an n power of n, so that is here's coefficient, and that in the most simplistic model can be viewed as n copies of the repressor X can bind to the promoter region of the gene Y cooperatively. Okay, if n is 2, then presumably 2 copies of the repressor need to bind at the same time for this to work. If n is 10, 10 and so on. Okay, so that is the idea. And I derived this form of expression basically, using DNA binding of protein and copies of protein collect cooperatively, and then as a function of protein concentration, you can estimate what fraction of the DNA molecules are free of the protein, because then proteins, DNA molecules, naked DNA molecules that are not bound by the repressor can recruit RNA polymerase and then transcribe mRNA at a certain rate of beta. In this case, the beta includes so this includes both transcription and translation. Okay, you need to make mRNA based on the DNA, and then you also need to make proteins based on that, and that gives you phenomenological production rate, beta, and that gives you the production of the protein. And K here can be called a binding constant. Actually, most common expression is a dissociation constant. And ideally, if the protein concentration is equal to K, then this becomes 1, and this effectively becomes 1/2, so that production rate of that Y protein will be half as maximal. Okay, so that is a concentration of the repressor at which the production rate becomes drops by a factor of 2, and this function generally looks like this. As a maximum value of beta at 0 repressor concentration, and it drops to 0 eventually. So today I want to think about, why it may be beneficial to have a beta, cooperative binding, and being other than a 1. If n is 1, then it's not cooperative. If n is 2, 3, or 4, it is cooperative. Is there any benefit to, controlling gene expression and regulating gene expression by having, cooperativity. Okay? So for that, let's start from a very simple system. You're making X, but there's no repression. Okay? So this means that if you plot how X is changing, and there is some that is the production rate, because now there's no repression, so that will be simply the production rate. And then there will be degradation given by alpha times X. So alpha is the degradation rate. So you're making the protein at a certain rate, but you're also degrading the protein at a certain rate that is proportional to its concentration. So at some point, you reach a steady state. Okay? And so in steady state, so this means that very simple. Okay. Now, but generally speaking, before you reach steady state, then this follows this function. Does that make sense? Yeah. So initially, when t becomes infinite, then this gives a steady state value. And then if you actually start the production, for example, at t equals 0, then that this gives you a 0 at the beginning. So if you start with nothing and then you turn on the production and wait, and then you plot the x as a function of time, then you'll see something like this. So x as a function of time, you start from 0, and then it'll reach that steady state after some time. And so you're interested in how quickly the system can adapt to the new equilibrium. And then let's define that equilibrium as T1 half, okay? To change by a factor of 2, half of the final change. So T1 half will be when this becomes 1 half, right? So this becomes log of 2 divided by alpha. So this is an interesting thing to remember. Basically, how quickly the level of protein can change is determined by one number. What is that? Protein degradation rate. Right? So that controls how quickly things change. It's not actually, even when you produce the protein degradation, when you start to produce the protein, what determines how quickly you reach equilibrium is actually not the production rate, it's actually the degradation rate. Right? Of course, the production rate will affect the steady state value. So this, I can call it simple regulation. Well, there's no regulation, right? You can just turn it on, turn it off, but there's no concentration dependent production rate here. Let's consider a system that is a bit more complex. You make X, but X is a repressor itself by itself and represses its own production. Okay? So that X then come here, bind here as N copies binding cooperatively to bind the promoter and to repress protein production. Okay, how do you write this equation? dx dt, rate of production of X. Actually, the time change of this guy will be beta times okay, I'm just taking it from this. You're using X to repress the expression. So this is the amount of repression you're getting times the basal production rate. Okay? And then we have alpha times X for its own degradation. Now what you want to do is to find out what the response time is. And how does it depend? Meaning response time depend on N. Okay, so I want to show that if you make N larger than 1, then response time can become much shorter than the protein degradation time. Okay? So that you can make the response faster by making, more cooperative system here. Okay, let's see how that works. I'll use one approximation. I'll assume strong repression regime. That is, there's already a lot of the repressor protein available in solution, much higher than this dissociation constant, so that this value is much larger than 1. Okay? Under this condition, X over K is much larger than 1 under strong repression condition. I guess I should just write this as auto repression. Then we can rewrite this equation as and I'm going to multiply everything by this. Multiply everything by this. If I do that, then I have so then I can make a standard substitution. So I can say define U as X to the N plus 1 power. Then this guy, and then du dt is right. Or dx dt is, sorry, or x times x. So if I then plug everything back in here, now this one takes the same form as that expression, right? Then you can solve for it. Right? So basically, u will follow a function that looks like this. Right? So let's do that. Steady state, then so that the function u will follow this type of shape. But now I can go back to x, which is u to the 1. So that is not my final result. Okay. So this tells you how quickly this steady state level of x can change as a function of time. So you can ask this question: okay, what is that? T1/2 for I put NAR for negative auto-regulation. So that is what I just discussed for negative auto-regulation function. And how does it compare to the half response time for simple regulation? Okay, that is, there's no concentration dependence, the dependence of the prediction rate. Then I have, this can be 0.006 for n equal 2. Okay? Just to have, n equal 2, just to go to dimers binding together, that makes the response time actually 15 times faster. And this can be actually, if you go to n equals 3, this becomes actually 50 times faster. Okay? So there is a merit in having cooperativity. You can make the adaptation to the new equilibrium so much faster. Questions? Yes?
So we use the strong repression approximation and also assume at time equals to 0, u equals to 0.
Well, so that is true. So this is not strictly correct, but I think it will be valid as long as you go beyond certain regime here, right? If you go beyond halfway point, then, yeah. So exactly by what factor you actually gain will depend on, on which approximation you use. But and the reason why you use it is because, of course, you can solve it analytically, right? If not, that's nonlinear equation to solve. Questions? Okay, so the homework problem that I just posted, one of them is following. The same situation, auto-repression, but now we actually look at cell-to-cell variations. Okay, you have the same, same everything except that different cells in your population can have a different beta value. So let's say there's a 10% variation of the production rate between different cells. But then, maybe you want to have all the cells have the same value, steady-state value of that protein, right? Then, some production rate changes between cells may result in actually in the steady-state value change. So basically, how does it work? So it turns out that if n is 0, if there's no concentration-dependent regulation, simple regulation, then whatever change that you get in beta between the cells will be directly reflected by the differences in the protein level in different cells. So if there's a 10% variation between different cells, protein level can be different by a factor of 10% between different cells. So noise is actually preserved. But if you have n equal, let's say 4, then 10% noise in this value between the cells is reflected only as 2% noise between the cells in the steady-state value. So higher n value, cooperativity also has the beneficial effect of reducing the protein expression noise between the cells, even when there are some underlying noise factors. Okay? So that is one thing that you'll look at in the homework problem. So now I want to go back to the toggle switch and then treat this as a mathematical problem and then discuss some of the solutions. So what is a toggle switch? If you want, you have x and y, and x is repressing y. The protein n copies, then binds to repress y, but y is also binding here to repress x. And I also use this type of notation to describe this. Right? X is repressing y is repressing x, and so when y level goes up, x will go down, x goes down, y goes up. So basically, once you start to go in one direction, you stay in that state, just like a switch of the lamp in the room. Okay, so the toggle switch. You don't have this intermediate state. Any intermediate state where x and y are equivalent should be unstable. Okay, so that's what you want to find. That's what you want to do. So then you can write things down in this manner: dx dt. So protein X is degraded at a rate of alpha. and then it is produced at this rate. So it depends on the repressor, its own repressor Y's concentration, N's power, and then binding constant, dissociation constant K. And likewise, what assumptions am I making here?
They're produced to decay at the same rates.
That's right. I'm assuming that the two proteins are made at the same rate, same basal rate, and the same degradation rate. And it's a simplification to make the math simple. But you can imagine that if you use different values, then it will still be the same. Just get different final numbers, but it is a useful approximation to make.
Also same.
That's right. I'm assuming that they have the same cooperativity factor of n, right? So it's, again, I'm just doing it, just to simplify the math. Right? What else am I assuming here?
Degradation rate.
Hmm?
The degradation rate.
Same for the two? Yes. Hmm?
Same K.
Same K, that's right. I'm assuming that the proteins have the same dissociation constant to their binding sites. And that's again an approximation. Okay? And one more thing that I'm assuming, which is a very subtle one, is that in reality, even when you have infinite repressor concentration, your production rate is not zero. There is some actually production rate, let's say, you can call it gamma, that is independent of the repressor concentration, because usually what they call this a leaky expression or leaky repression. You have repression, but it's a little bit leaky. So even when you have, a cell completely filled with a repressor, you can still get some transcription going. But this is fine. Okay? And I can simplify it further. I have to change the variables, and it becomes that's gamma here. Okay? Suppose U and V are positive numbers because they're concentrations, and or zero. Now, in steady state, or if you have, let's go this way, U0, V0. So in the steady state, you can determine the stationary point U0 and V0, where the left side becomes 0 because nothing changes there, right? So this means that U0 is equal to So when in the steady state for the stationary points, then these two equations will be satisfied, right? Because this is 0 and that is also 0. So let's try to solve it just graphically. So let's plot this function. V equals gamma of 1 plus u squared. So when u is 0, this becomes gamma, right? It starts from gamma here. And then when u becomes infinite, then this becomes 0, right? So actually what you get is a function that looks like this. I can do the same, plotting u as a function of v. Use that. When v is 0, u equal gamma, and when v is infinite, this becomes 0. So I can do the same here. Okay, and then goes like this. Okay, and where are my U0 and V0s? When these two curves meet each other. Okay, so these are my solutions. These are my stationary points. Here, here, and here. Okay, so among the 3 points, which one will be my desired on and off stage for a toggle switch? B, when your B actually turns out that U and V are identical to each other, right? This equation is symmetric. When U equals V, that's actually that's as if you're holding the light switch right in the middle position, which is the unstable state. Okay. So A and C are. So if you look at it from the viewpoint of U, production of gene product U, then this will be unstate. For U, or if you want X, and this will be the off state for U for X, because the protein level is high here and level is low. Okay. So the A and C are the stable stationary points. and B is the unstable. And that's actually, when you design the system by tuning the parameters, you want to make sure that these become stable and this become unstable. Okay? Okay, what do you mean by stable? What I mean by this, if somehow there's fluctuation in the protein level in the cell, inside the cell, so this becomes kind of shifted somewhere here, away from that stationary point. But then, quickly it'll have to decay back to the stable stationary point, right? So that's what I mean by a stable point. Whereas if you make a small deviation from this one, just like when you're trying to hold the light switch in the middle and then you just let it go, then any small deviation from that midpoint will flip the switch to the on or off position, right? So any small deviation can actually amplify exponentially. Either can go here, or can go there. And depending on the parameters, it can circle around to move away, right? Or you can just go there directly. That depends on the eigenvalues of the matrix that you will deal with. So that is actually a graphical explanation of what's going on in this toggle switch. Yes?
It seems like you could get just one solution if you get the right value of gamma. You don't have all three intersections.
Okay, explain.
If your blue curve was, if that initial shoulder as V increases were longer, you wouldn't intersect at A or P, you would just have the intersection at C.
Oh, so if this guy somehow drops down here. Oh, sorry, drops down here.
Yeah.
Is that possible?
I guess if gamma
Not when you make the problem symmetric.
Okay.
Perhaps if you don't make it symmetric, it's possible, right? There's no physics here, it's just mathematics. I would say it's pretty elegant mathematics. Right? You can actually use a linear algebra to understand. So, how to describe the system? All right, so let's look at this more mathematically. The general expression that I can write down is that the UDT is a function of U and V, F1. So that is the most general form. And the VDT so in so when you evaluate these functions at the stationary points, then this becomes 0. Okay. But now, if that is your stationary point, U0 and V0, then what happens if you deviate slightly from that point? Okay. So let's make a small deviation. So now u is u0 plus delta u. And delta u, this can be a function of t. It can be a function of t. And this is not a function of time. So it's a constant. But this can be a function of t. Same for v. Then dot is a time derivative, yes, and because if you take the derivative of the constant, that becomes 0, so you only retain these two, but that is equal to what's shown here. So what you're getting here is well, I simply replace u and v by, this expanded form around the stationary points u0 and v0, assuming these numbers are small. And then this becomes value of these functions themselves at the stationary points plus everything. So I simply expand these functions this way into matrix form. And then just based on the definition, this becomes 0. So what you end up getting is this equation, basically deviations around the stationary points will have the time dependence given by equation. And the solution So it'll take, generally this form. The first eigenvector time is the exponential to lambda 1t. Lambda 1 is that corresponding eigenvalue, plus eigenvector 2 times e to the lambda 2, and with some constants here and there. Okay, so that is the function. Now, what you can realize is that if any of the two values is larger than 0, right? Then this is a function that grows exponentially, right? This means that if you have a small deviation around that saturation point, you don't go back to 0. You actually go far away. You move away from that point. So that makes that point unstable. In contrast, if both numbers are negative, right? What does that mean? That any kind of deviation will decay exponentially over time. So you go back to the stationary point. So basically, you want to tune the parameters so that if this is what you want, then these points, eigenvalue should be negative, and here eigenvalue should be positive. Okay. Of course, eigenvalue can also be imaginary, right? So then, if you have a negative real number plus an imaginary term, then you get an oscillation. Basically, if you have a negative real number in the eigenvalue and the imaginary number as well, then you'll spiral to go back to the station point. If not, you will spiral out. Okay, but that's the idea. Okay, and you guys know how to determine the eigenvalues for this, right? I forgot, but.
You go to MATLAB and type in I.
Really? MATLAB doesn't do that if you give you the symbolic expression, right?
I think it's have numbers.
Yeah.
Attica does.
Or you can ask your friend. That's what I used to do. All right. Any question? Okay. We'll take a break here, and then I'll come back to discuss some additional topics. I want to just quickly review what we discussed last time. This 2005 paper measuring gene regulation function at the single cell level experimentally. Just to review, as you increase the repressor concentration, production rate of the target protein will go down, and what is the functional form, that is the question. So what they did was to tag the protein that you are studying with the green fluorescence protein, colored in green here, and then they also tagged the repressor with a red fluorescence protein. And you start with larger repressors in the cell, so there's no green at all. And then as time goes on, the cells divide and divide, diluting that repressor. And then once the repressor concentration becomes low enough, then the green proteins start to get produced. And that's why you see something like this. Okay? Of course, you can estimate the protein concentration of the red kind repressor by measuring the intensity of the red fluorescence. You cannot use the intensity of the green directly as a measure of the production rate, because this is a time-dependent change. So here, to get this, you have to measure this at 2 different time points, and then, of course, take the difference and divide by the time difference to get the production rate. But anyway, that's how they did the measurements. And I show you 2 movies. Just for fun, I show you one of them. Okay. This is the reason why I like the papers by this group, because they always include colorful movies. And I discussed the next paper. They usually do not show any movie. So although their work is also very interesting and important, somehow I liked movies. Anyway, do you have a question? So you can quantify the data, and these are real signals. This is one example where, when cell divides, protein level drops by a factor of 2. This is log scale, another factor of 2, and so on. And then green level actually then shoots up. You can see that there is some production as soon as that red level goes down a little bit, right? And then, but cell divides, it goes down, and then finally it shoots up. So there is an anti-correlation, as you expect. And again, the problem was that they didn't know how to, there was not a really good method to estimate the absolute concentration of the protein. So they use the fact that when cells divides, then how much of that red goes to one daughter cell versus the other daughter cell will depend on that there's a noise in partitioning, but partitioning noise will depend on the level of the protein. Again, if you have 1 million protein, then fractionally that noise of partitioning will be very small. But if you have 10 proteins that you're dividing into 2 cells randomly, then you'll have a large amount of partitioning noise. Right? So they use it to estimate the absolute number. You will discuss this in one of the homework problems, but in the end they were able to estimate the production rate as a function of the repressor concentration, still apparent concentration, and then they use the function that we discussed today that has this functional shape to fit the data, and they obtained 2 key parameters. One is N, Hill coefficient, close to 2 for both of them, and then this is the dissociation constant at which you obtain the half maximal repression. Okay? So that is that paper that you consider in your homework problem. So you might ask, because we discussed single molecule fluorescence measurements in this course, and I also told you that you can look at single protein molecules or complexes and measure photobleaching profiles of single complexes to deduce the stoichiometry, how many copies of proteins are there, and so on. But so why not just, count individual proteins instead of doing this binomial distribution analysis? The problem is that there are actually too many in the cell. If you have, a lot of proteins, you cannot see them as individual proteins. And okay, so then maybe you measure the intensity of one protein at low expression level and use it as a calibration, and then convert the total intensity of the cell to a number of proteins. But usually there's just too much background fluorescence in the cell to allow single molecule detection in many cases. So a year after that Elovich paper, a group in Harvard University actually showed that now you can measure the production of a single protein, birth of a single protein inside a living bacterial cell. So I'll discuss that paper next. Oh, that's my friend, Sunny Z. So this paper is titled Probing Gene Expression in Live Cells, One Protein Molecule at a Time. And let's see. Okay, you see two different things, right? First, this lab does not, back then, did not have a good automation system to correct for drift of the microscope. So over several hours, you see a lot of drift, okay? In the Elovich paper, you don't see that because they have a good correction system. Second point is that you see flashes of a yellow light as the cells grow and divide. So those are corresponding to single protein molecules that are being produced, okay? And then photobleached. Yeah. And let me show you another example here. Okay, so that's what they claim to have done. They can detect every time a single protein of certain kind is produced in the cell. Now, how did they do that? So here is the experimental system. So this is the E. coli cell, and E. coli has two membranes, inner membrane and outer membrane. Okay. And there is a DNA they put into the chromosome of the bacterium. And then there is a RNA polymerase making mRNA. The ribosome is binding to the mRNA to make a protein coded by the DNA region here. And the DNA sequence was chosen such that first you make actually, the RNA polymerase binds here, promoter, and then it goes in this direction, right? And so RNA polymerase can bind when the repressor is not bound to this one, and then start transcription of the mRNA, and then ribosome will make that into a protein. They put two artificial sequences here. One is called Venus, shown in green. Actually, Venus is one of the fluorescence proteins that people engineered. So that when the protein is made, it can fold and process quickly to give you fluorescent signal within just a few minutes, like 7 minutes and so on. Typical fluorescence protein that you make, it takes over an hour before the protein is produced until you get, you make the protein by the ribosome, but then some chemical reactions have to occur for the protein to mature as a fluorescence protein. typically takes more than an hour. It takes actually several oxidation reactions with oxygen molecules in solution. But they found the mutant, they named Venus, that you can actually fold and mature. They can mature in just a few minutes. And if they want to measure the protein production as a function of time, then you don't want to wait until this protein matures, because then it adds additional uncertainty about the production timing. So that is one reason why they picked this particular variant of the fluorescence protein. And TSR is a sequence motif that is found in some of the bacterial inner membrane proteins. So if you connect the fluorescence protein to that TSR sequence, then as soon as the protein is made, then it'll actually go and bind to the cell membrane and be localized there. Why? The reason is the following. So if you have a GFP Venus only made in solution, then inside the cell, this will diffuse inside the E. coli body very rapidly. And diffusion is actually too fast for you to see a single molecule. So when you look at this using light microscopy, then this guy just signal is spread over the entire cell body, and it's hard to distinguish that from the cellular background fluorescence. But once a protein binds to the cell membrane, it can still diffuse in the membrane, but diffusion is, I would say, about 100 times slower. Okay? Diffusion coefficient is about 100 times lower. So then you can actually see it as a single fluorescent spot. Okay? And that way you can beat the background signal.
Why did they have black wine after?
Oh, so that was there was nothing special about this. LAC-Y is lactose transporter. Okay, so that is a natural part of that LAC operant that is found in E. coli. They simply inserted this into the LAC promoter region, because they wanted to study a natural promoter in terms of noise in protein expression and so on. Any other question? Is there anything else that is unclear here on this slide? Okay, what is polypeptide?
It's using chain of amino acids.
That's right. And why don't they call it a protein instead of polypeptide?
Often they're cut into smaller pieces and make individual proteins.
That's right. So this motif is not actually an entire protein. It's a polypeptide, a part of a protein. Right? So you just get that signal, membrane localization signal peptide used here. Now I think I explained every word shown here, this line. Okay? So the idea is that normally LAC repressor is bound here, so protein is not being made. And occasionally this guy, kind of get distracted, takes off a little bit, and then the RNA polymerase can bind here and then, make a copy of mRNA. And then this guy rebinds, and then RNA will be made into proteins. Okay? So the question is, okay, how many mRNA molecules will be made per event, per LAC repressor dissociation? And then how many proteins are made per event? Okay? So you have, by the way, this is just demonstration that they can do single molecules. So this is the regular light microscopy, no fluorescence, just showing two cells, and then they can see a fluorescent spot here, while this one doesn't show any fluorescent spot, just autofluorescence, cellular background autofluorescence. If you take a cross section along the cell body, then you see that this is a signal level from the microscope slide, and then this is the level of background fluorescence from the cell itself, and then you have these two peaks coming from two single proteins bound to the membrane. And they can also make that Venus protein and stick it to a glass cover slip and measure the intensity of fluorescence of a single protein as a function of time. They see single-step photobleaching, and they can determine that intensity level as a calibration. And then when they compare the intensities of these guys versus this one, they see a pretty good agreement. So that's what they use as evidence that they are seeing single protein molecules. Okay, so I showed you a movie. Cells divide, forming a colony, and then you see flashes of light. So that's presumably when a single protein is produced. And then you can actually form a lineage tree starting from one cell, divides into two, daughters and granddaughters and so on. And for each cell, you can trace, which lineage it follows, And then for each lineage, then you can actually plot every time, you see a fluorescence signal. So basically, for this lineage of cell, then you see like 4 clusters of fluorescence signals. And here you see 5 clusters and so on. And you can notice that just from just looking at it, it doesn't look like the signal is happening randomly. They appear to be clustered. And you can also do mathematical analysis to show that this is actually not just random events that appear to be clustered. They are really clustered. So what they were able to claim is that proteins are made in bursts. So this was the first real direct demonstration that proteins, when they are made, you don't make just one protein and turn off that protein expression. You can make several proteins in a burst.
But this is natural. Yes, because if the repression factor is dissociated, there is some time for it to make several proteins instead of just one.
Yes.
Is this intuitive or counterintuitive?
Oh, well, I mean, it makes sense. It makes a lot of sense, right? Because it's already known for many years that single mRNA molecule can be bound by multiple ribosomes, right? So even if you make one mRNA, based on that you can make several proteins, right? So that makes sense. But really, there's no real direct demonstration that this can be done in this way inside the cell. Actually, whether proteins are made in bursts or large bursts or small bursts has some implications on gene expression noise. For example, cell-to-cell variations in protein levels, even when other conditions are identical. So let me give you a reason for that. So there's a really famous paper by Alexander van Oudenaarsen's lab. Back then it was in MIT, but now he's in the Netherlands, directing a large developmental biology center. So this paper in Nature Genetics is titled "Regulation of Noise in the Expression of a Single Gene," and there's no actually colorful picture movie in the paper. But despite that, it is a very important paper. I believe that the yeast strains that they engineer in this paper are still the most frequently requested material from the lab. So let's consider the same kind of analysis. So you have a DNA, and then you make an RNA with a certain rate. The RNA can degrade at a certain rate, and then can be made into protein at a different rate, and protein can also degrade. Okay? So let's imagine that using you can actually use this to simulate the copy number of the protein as a function of time, right? And so that's the simulation. Protein number as a function of time. You can see that number is fluctuating because it is a stochastic system, right? Fluctuating. But this simulation at the bottom was done with B equal to 1. B is the burst size. When a protein is made, this one assumes that you only make one protein per production event in the stochastic modeling. Whereas in this top case, B is 10. So you use a parameter so that on average, 10 proteins are made per burst. And you can see that as a function of time, there is a much larger noise, right? When you have a large burst. I mean, intuitively it makes sense, right? But what it also means is that, instead of measuring this as a function of time from a single cell, you can actually measure many, many cells, but at a given time point. That means that then if you look at 1,000 cells in a population at a fixed time point, then they will also have variations in the copy number. Okay? So compared to this case, the small burst versus large burst, the large burst will give you larger cell-to-cell variation of the protein level. So in this paper, they couldn't measure this in a single cell as a function of time, right? This is a simulation. But what they were able to do is to measure the snapshots of many, many cells at a given time point by measuring the protein level using fluorescence. Okay? Your protein is now fused to a fluorescence protein, and then you use a device called flow cytometry to determine the protein copy number, and they were able to say something about the system. So I'll come back to this one on Thursday, and the remaining lectures, I decided to talk about modern gene expression analysis tools. So there are actually very new technologies that are really, really popular to analyze gene expression at the single cell level, at the genome-wide level. RNA-seq is one, microarrays and ribosome profiling. So I'll try to explain some of the most exciting topics that are very popular and hot these days in the remaining lectures. I have four more to go. Okay, thank you.